Hello everyone, my name is Soham Jha. I'm a first year MBBS student from GMS Medical College, Sola, Ahmedabad, and I'm very grateful and thankful to the Make Medical Easy committee for allowing me to present a series of lecture on general metabolism. And today we will be starting from on carbohydrate metabolism. The topics today, which will be covering are digestion and absorption of carbohydrates and related clinical application, glycolysis, CORI'S cycle, and glycogen metabolism. So now we begin. Digestion and absorption of carbohydrates and its related clinical application. Carbohydrates are present in complex polymers which are required to be broken down to simpler parts for absorption in their metabolism. हम लोग जो इंसान होता है, we the humans, we cannot actually metabolize directly the complex polymeric form of the glycogen, not the glycogen, but complex polymeric form of the carbohydrates, which needs to be broken down into smaller part for absorption and digestion and absorption, and its metabolism. It can be done via the mechanically, which we chew, and chemically by the action of enzymes. And initially, we do via chemically by heating or by thermal breakdown of the carbohydrates. And further on, as we all know, the digestion of carbohydrates begins in the mouth. The first thing which happens in the whole digestive system is chewing of the food associated digestion of the carbohydrate. So in mouth, it begins with the action of salivary alpha amylase. Then comes the digestion of carbohydrates in small intestine, which occurs in duodenum by the action of pancreatic amylase, second part of duodenum. And then comes action of intestinal juices, which are present in the small intestine. Further on, acted upon by the maltases, sucrases, isomaltases, lactases, which are the type of disaccharidases and trisaccharidases actually. And then comes the clinical application of lactase. Lactase is actually an enzyme which is within small intestine, produced by the brush border cells of the intestine, enterocytes. The lactase is actually involved in the breakdown of lactose into glucose and galactose. Absence of lactase enzymes causes accumulation of lactose in the bowel, which is later on excreted out. And before its excretion, it is acted upon by the certain organisms, microorganisms present in the large intestine, which digest lactose and derive energy from it. And as they proliferate, they produce certain toxins and certain gases, which causes flatulence and increase in intestinal motility, which could cause loose motions. The main reason for deficiency of lactase enzyme is it can be congenital by birth defect, or due to age, or due to reduced transcription of the enzyme. The reason behind the reduced transcription is that the enzyme actually is regulated. The production of enzyme is regulated by the amount of lactose which is consumed by the person. So abruptly or suddenly the consumption of lactose is stopped, which actually leads to reduced lactase enzyme production. Therefore, as if the person again abruptly continues the consumption of lactose, might lead to lactose intolerance. And in case of aging, in old age people, lactose intolerance is seen as production of lactase enzymes is reduced. So it is seen in old age also. The treatment RX is, as mentioned in the PPT, is lactose-free milk consumption and avoid direct milk products, and consumption of curd. Consumption of curd, as actually curd contains lactobacillus, which helps in partial digestion of the lactose, and avoiding direct milk products, because the direct milk products contains a larger concentration of lactose. Therefore, it might be harmful if lactose is consumed in a larger amount by a person who has lactose intolerant. And lactose-free milk, presently, it has been used for the last 5 to 6 years. It has come in a view of many people. Therefore, consumption of lactose-free milk is seen. Then comes the transport of glucose. Transport of glucose or transport of carbohydrates, I should say, We will mainly focus on transport of glucose, occurs mainly via facilitated diffusion, by action of different types of transporters. These transporters are known as glucose transporters, or simply GLUT. There are some of different types of GLUT, actually S-GLUT, uh, S-GLUT1, S-GLUT2, and other different forms. GLUT1, GLUT2, GLUT3, GLUT4. These are simple glucose transporters. S-GLUT is sodium-dependent glucose transporter, which absorbs glucose along with sodium ions. Clinical application is administration of intravenous glucose dextrose solution along with diluted saline. Usually we don't require to do this, but if associated iron loss and associated electrolyte loss and fluid loss is seen, therefore intravenous glucose along with electrolytes and water is administered for rapid rehydration and compensate for the compensation of iron loss. In case of diarrhea, actually we do in case of diarrhea, we give ORS, oral rehydration therapy. So that ORS therapy, oral rehydration solution therapy, in which compensation of fluids and electrolytes is done by administration of ORS. ORS basically containing salt, normal table salt and sugar along with lots of water. So and if you add lemon to it, then it becomes nimbu pani, which we use in normal cases. If a person is suffering from weakness, actually, if a person is suffering from dehydration due to peak stomach, we provide nimbu pani or ORS. Actually, it is a kind of ORS. Nimbu pani is a kind of ORS. Okay? And then comes the GLUT2. GLUT2 is a simple protein which is involved in facilitated transport of glucose, uniport, functionally involved in homeostasis of blood glucose via its change in rate of transport, which influences the function of pancreatic hormone and enzyme secretion. Actually, what happens is that GLUT2 is present in different sites like intestinal epithelial cells, liver cells, beta cells of pancreas and kidney, and in kidney cells. So what happens is if the amount of glucose present in the food increases, then the amount of glucose absorbed by GLUT2 increases, which directly influences the release of different enzymes and insulin, which causes increased insulin secretion or decreased insulin secretion as per the body's status of glucose. Okay, so this is what happens when the GLUT2 has changed in rate of transport of the glucose. If the rate of transport increases, then amount of glucose would actually increase in the blood. So the cells will say, let's increase the insulin production so that we can absorb some much more amount of glucose. So say that we absorb a lot of glucose, then what should we do with the glucose? The glucose either goes for glycogen synthesis or glycolytic pathway or any other further production in glucogenic amino acid production. There might be a chance that the byproduct which is formed could be going over there. Okay, and GLUT4 is a major glucose transporter for adipose tissue and skeletal muscle. In case of DM2, function is impaired. DM2 is actually diabetes mellitus type 2. The number of GLUT4 transporters is actually changed by the insulin. Insulin actually acts upon the cell, which causes the increase in number of GLUT4 channels on the surface, which increases further glucose absorption and uptake. And there's a special point that GLUT5 is a fructose transporter. And now we come over to glycolysis. Please read thoroughly in the initial part because this part can be asked for short questions and for Viva also. The clinical application will be used in the further clinical scenarios also. Glycolysis, also known as EMP pathway, and responsible for conversion of glucose to pyruvate in case of aerobic respiration or lactate in case of anaerobic respiration. Significance: primarily pathway for glucose metabolism, solely responsible for energy production in case of anaerobic condition in skeletal muscles and anaerobes also, anaerobic organisms also. Byproducts are involved in production of various amino acids fats, some of the parts of the fats and intermediate products which are linked to gluconeogenesis. And the total number of steps are 10, and the irreversible steps are 1, 3, and 10. So here we can see the glycolytic pathway. The first step is phosphorylation of glucose, which is done by the action of hexokinase enzyme, done via use of ATP. The ATP is broken down to ADP and released, and glucose 6-phosphate is obtained. Then the first step is irreversible. Actually, here we can see by also the arrow shows that the reaction goes in a single direction. Therefore, this is a irreversible step. Then comes the second step, in which glucose 6-phosphate is converted into fructose 6-phosphate by the action of phosphoglucoisomerase or phosphohexoisomerase. This reaction is also readily visible. And the further comes up the step 3. The step 3 is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate has one more ATP as compared to the initial part, the substrate. So there will be utilization of ATP. The ATP will be converted to ADP. Adds phosphate and prepares the fructose 1,6-bisphosphate bisphosphate for cleavage. Now the fructose 1,6-bisphosphate goes for the cleavage. The cleavage occurs, which produces two products. Those are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Those can be interchanged. So what actually we do is that dihydroxyacetone phosphate again converts into glyceraldehyde 3-phosphate, which participates in the next reaction, which is conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, which occurs via the action of glyceraldehyde 3-phosphate dehydrogenase, and one ATP is produced over here also. Then comes the next step, in which ATP is produced, in which 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate via the action of 1,3-bisphosphoglycerate kinase enzyme. Further on, 3-phosphoglycerate is converted into 2-phosphoglycerate. Phosphoglucomutase is the enzyme which is involved in this conversion. Then the phosphoenol pyruvate is produced by the action of enolase enzyme. Then comes a step in which phosphoenol pyruvate produced is converted into pyruvate via action of pyruvate kinase, which converts ADP to ATP, therefore producing another ATP. And then comes the fate of the pyruvate. So what will happen next to the pyruvate? Actually, I will be discussing more about this. We can see the preparatory phase and the payoff phase. The preparatory phase is the initial phase in which the glucose is prepared for the metabolism, in which phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate, as mentioned over here, is we can see. In the preparatory phase, we actually utilize ATP. So we can say that this is preparatory or the investing phase of the glycolysis. Then comes the payoff phase, in which conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of ATP. So we can see that we obtain our NADH to our ATP. Here in this case we see total 4 ATPs are obtained along with 2 NADH2s, because the number of glucose molecules may have changed. There might be a reason. And then comes the fate of pyruvate. The fate of pyruvate depends upon the ATP and the body's requirement, what is actually going with the body. So glucose is converted into 2 pyruvate molecules by 10 successive reactions. In case of anaerobic conditions, alcohol formation by the fermentation process in yeast. The yeast carries out fermentation process, actually produces 2 ATP molecules. Not that necessary to be remembered, but it occurs as a fact. You may please remember it. it could be asked in the Viva. Or further, in aerobic condition, by the oxidation aerobic condition, we see in the production of 2-acetyl coenzyme A, and we see two CO2s are removed, means two carbon molecules are removed. So we started with 4 carbon molecules, then we have two molecules of same type of 3 carbons, then 2 CO2s are removed. Therefore, we have 2 molecules of 2 carbon containing molecules, and which undergoes citric acid cycle. It will be discussed in the next video. I will explain further upon it. And it produces 4 CO2 molecules and 4 H2O. This process, the citric acid cycle, is main process which produces the converts the products of glycolysis into the major energy products like ATP. We actually see the total energy produced in the form of ATP. So the amount of ATP produced is seen finally counted from the end of citric acid cycle. And the citric acid cycle is done by the animals, plants, and the many microbial cells under aerobic condition. All higher level animals and plants usually carry out citric acid cycle for energy production because it is much more efficient as compared to other minor pathways. And in case of anaerobic condition, as I had talked before, for the fermentation, there is another anaerobic condition which is seen in microorganisms as in case of gut bacteria, in which the product is 2-lactate. Fermentation to lactate in vigorously contracting muscles is seen, and in humans also it is seen, in an erythrocyte and in some microorganism is also seen. So this is the fate of pyruvate. And energy yield, it can be seen as over here. Glyceride 3-phosphate dehydrogenase 2-NADH etc oxidative phosphorylation, number of ATP synthesized is 6. Phosphoglycerate kinase substrate level phosphorylation, the number of ATP synthesized is 2. Pyruvate kinase substrate level phosphorylation, number of ATP produced is 2. 2 ATP are consumed in the reaction catalyzed by hexokinase in phosphatidylkinase, which we had seen over here in the preparatory phase. We see over here that in the preparatory phase 2 ATP molecules were consumed. So over here minus 2 ATPs. And phosphatidylkinase does not require any ATP over here, so 0. Then the net ATP synthesis comes out to be 8. Pyruvate dehydrogenase PDC complex, which is also seen. I will further explain in the next video. 2-NADH and ETC and oxidative pathway, 6 ATPs are obtained in citric acid cycle, which will be explained in the further video. But the energy I will explain over here is by the action of isocitrate dehydrogenase 2-NADH ETC oxidative phosphorylation, 6 ATPs are obtained. Alpha-ketoglutarate dehydrogenase 6 ATPs are obtained. Succinate thiokinase substrate level phosphorylation, 2 ATPs are obtained. Succinate dehydrogenase 2-FADH ETC oxidative phosphorylation, 4 ATPs are obtained. Mannate dehydrogenase 2-NADH ETC via ETC oxidative phosphorylation, 6 ATPs are obtained. Total ATP per mole of glucose under aerobic condition is 38, and total ATP per mole of glucose under anaerobic condition is 2. And also there is a point which has recently been added that 6 ATPs are produced if NADH use malate shuttle. Only 4 are produced if glycerol phosphate shuttle operates, in which case total ATP synthesized per mole of glucose is 36 and not 38. So this correction has to be made. And while explaining, I also realized that the whole glycolytic cycle and the biochemical reactions are actually very hard to memorize. So what I used to do is I actually used to make one cycle for our exams. For the exam purpose, I will be explaining that, because it will be very helpful for students like me and other students for exams. That what I used to do is make the whole cycle in a plain page. I am presently using a A4 plain paper, and I make the whole cycle. And I explain everything related to the particular reaction. If I am talking about the first step, hexokinase, then there are different types of two enzymes of types of hexokinase. Hexokinase and glucokinase is also seen, which are present in different tissues. and I used to write everything about the particular reaction in a particular corner, so that about the 10 reaction, I have 10 corners in a single page, and I can look up to the whole reaction, and I can easily revise every single day about in 2 to 3 minutes. So that will be very helpful for the other students also. From my side, as a suggestion, if you have a better method, please suggest in the comments, because that will be helpful for me and other students also. Next comes the regulation of glycolysis. We can see the action of G6P, glucose 6-phosphate, is negative over hexokinase. So it actually prevents production of or I should say uptake of glucose, metabolism of glucose, and glucose 6-phosphate phosphatase actually is promoted by the action of G6P. Glucose 6-phosphate actually enhances the action of glucose 6-phosphate. Then comes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, which is actually fructose 2,6-bisphosphate is promotes, and AMP also promotes, because actually ATP is consumed over in this reaction, and ATP inhibits, and citrate also inhibits this reaction. And in the reversal of this reaction, in case of glycogen synthesis, fructose 1,6-bisphosphatase is inhibited by fructose 2,6-bisphosphate and AMP also, iodine monophosphate. Further on, and in case of action of pyruvate kinase for conversion of phosphoenol pyruvate to pyruvate, the fructose 2,6-bisphosphate AMP have a positive allosteric effect over pyruvate kinase, and the negative allosteric effects of ATP, acetyl coenzyme A, and alanine is seen over the pyruvate kinase. And one of the reverse reactions, that pyruvate is converted to oxaloacetate by the action of pyruvate carboxylase. There is a positive allosteric effect of acetyl-CoA over this reaction. So now we come over CORI'S cycle. It is an important Viva question. It can be asked as a short question also, 1 or 2 marker, because it is required. It is an important cycle. It has some clinical, not much of a clinical importance, but it could be asked. It is also known as lactic acid cycle. What happens is, in case of anaerobic, glucose is converted to pyruvate acid and lactate. It is not completely oxidized to CO2 and H2. So because of the O2 deficiency, what happens is, due to strenuous muscular activity, the oxygen present bound to the myoglobin is all exhausted. So what happens finally is that lactate accumulated in the muscle is then transported to the liver, where the lactate is converted into pyruvic acid. It is actually, lactase is recycled, and it undergoes gluconeogenesis, in which by the action of 6 ATPs, glucose is formed, and the glucose is again via blood transported to the muscles and reutilized by the muscle cells. Here we can see the selfless action of the liver. Okay, we can see the selfless action of the liver, by which the liver provides the glucose produced by itself by the energy utilization. 6 ATP is a large amount of energy for cell. 6 ATP is actually energy currency, so we can say 6 ATP is a big thing. And the glucose, for the glucose production, the liver itself is doing so much and providing energy. So liver acts as a selfless organ. And the significance of CORI'S cycle is that it helps in utilization of lactate, product of anaerobic oxidation, provides glucose to cells so that it provides energy to the cells, prevents accumulation of lactate in the muscles. So what happens is that if the lactate accumulates in the muscle, it causes cramps and pain, which is usually seen in case of very high strenuous activity. If a person runs for 2 kilometers continuously, then he or she might feel cramps if they might stop. Okay, so what happens is, the prevention of lactate accumulation is done by CORI'S cycle. Now we come to the glycogen metabolism. Glycogen is nothing but a glucose polymer. Glycogen is seen mainly in animal kingdom, and in plant kingdom we see starch, polymer of carbohydrates. commonly seen for the storage in plants is starch, and in humans is glycogen. Synthesis of glucose from non-carbohydrate substrates is known as gluconeogenesis. Glycogen metabolism has certain parts that synthesis of actually I should have written glycogen over here. It is glucose, sorry for that. But what happens is the process is known as gluconeogenesis. Major substrate which undergoes this type of reaction are lactate, which I had mentioned earlier. The lactate was actually going for gluconeogenesis, as mentioned over here clearly. Amino acid, certain glucogenic amino acids, glycerol and propenyl CoA. Propenyl CoA is a byproduct of fat metabolism, which I will discuss further in the upcoming videos. Regulation occurs via allosteric modulation of pyruvate. It is pyruvate, not carboxylase, via concentration of acetyl coenzyme A, activation of citrate and deactivation of activation by citrate and deactivation of fructose 1,6 bisphosphatase by the action of AMP, and ATP and hormonal regulation. Glucagon and glucocorticoids have enhancing effects on gluconeogenesis and depressing effects on glycolytic enzyme. Okay. Glycogenolysis, in which degradation of glycogen by action of various enzymes occurs for obtaining glucose from glycogen, is known as glycogenolysis. Action of glycogen phosphatase along with PLP pyroxyl phosphate as per enzyme acts on alpha-1,4 linkage by cleaving it and produces levothyroxine. Levothyroxine is nothing but a small protein molecule which is present in the center of glycogen molecule, which helps in binding the glucose chains, 2 to 3 glucose molecule or longer glucose chains. This leaves a unit of 2 to 3 glucose molecules together, further broken down by the action of alpha-1,4 glucose transferases. It is not glucagon, it is glucose. I'm sorry for the mistake again. Which free this 2 to 3 glucose molecules. Then alpha-1,4 glucosidases debranches further from alpha-1,6 linkages and the free glucose is released. The enzymes transferases and alpha-1,4 glucophosphorylases converts the branching point into a linear one and produces glucose small chains of glucose molecules. Phosphoglucomutase converts the glucose 1-phosphate product of previous step into glucose 6-phosphate so that it can be acted upon later by glucose 6-phosphatase in liver. Actually, glucose 6-phosphate production is a very crucial step, or it can be say a tactical step by the cells, so that either it can be trapped in the muscle or it can be only removed from the liver for as in the form of glucose for the cells to be metabolized. Okay, so it is a very essential step. And only liver has this ability to release the glucose in free form in the blood, whereas muscle cells do not have this particular enzyme, which is the reason why muscle glycogen is not responsible for maintaining blood glucose. Glucose in form of glucose 6-phosphatase, glucose 6-phosphate is directly utilized in the glycolytic cycle via the second step of glycolysis. 3 ATPs are obtained via glycogenolysis, as there is no initial utilization of ATP for phosphorylation of glucose. Actually, we use isomerases for transfer of phosphate group. Therefore, no ATP is required. So, also here we can see the relation of glycogen metabolism to various other steps that here the glycogen breaks down by the action of glycogen phosphorylase. Glucose 1-phosphate is produced, which is acted upon by phosphoglucomutase, which breaks it down again to glucose 6-phosphate. The glucose 6-phosphate is also derived from the glycolytic cycle by the action of hexokinase over glucose, and there is an ATP involved, first step of the glycolysis, which is irreversible. Now the glucose 6-phosphate is undergoes a reversible reaction by the action of G6 PI, or I should say glucose 6-phosphate isomerases, which converts glucose 6-phosphate to into fructose 6-phosphate, which is then into which undergoes a reversible reaction. It goes backwards by the action of fructose 1,6 bisphosphatase isoenzyme to form glucose 6-phosphate from fructose 1,6 bisphosphate. Or either it goes into the forward direction by the action of 6-phosphofructokinase, which is active on glucose 6-phosphate, produces fructose 1,6 bisphosphate. Now the reaction, next the reaction is that Fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate by the action of bisphosphate aldolase. Or it can be converted into dihydroxyacetone phosphate by other reaction and into triose phosphate by the action of triose phosphate isomerase into 3-glyceride 3-phosphate, and it undergoes again a reversible reaction by the action of glyceraldehyde 3-phosphate dehydrogenase. So we can see over here dehydrogenase. It removes water molecule, so it is dehydrating, actually dehydrating the molecule. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. So there is also an addition of another phosphate group. So there must be an involvement of 18. So the trick which I am using for explaining the chart could be used also for memorization. Actually, this is a good way, because via just remembering the products and the substrates, we can also remember the enzyme and if there is requirement of energy or not. So please keep this point in mind. And I am going further over the reaction is that the 1,3-bisphosphoglycerate is acted upon phosphoglycerate kinase. Kinase is a type of enzyme, as we know, has a relevant function, which converts into the 1,3-bisphosphoglycerate into 3-phosphoglycerate. Again, some similar steps can be seen that 3-phosphoglycerate is converted into phosphoglycerate mutase by the action of phosphoglycerate mutase into 2-phosphoglycerate, and beta enolase converts into phosphoenol pyruvate, and then again acted upon by the action of pyruvate kinase isoenzyme M1/M2. There are two isoenzymes over here, and it converts into pyruvate. The fate of the pyruvate now again can be derived from over here. Either the pyruvate goes for pyruvate dehydrogenase reaction PDHC complex, or it can undergo further reactions for fatty acid synthesis or amino acid synthesis, glucogenic amino acid synthesis. Now here you can see glycogenesis. Actually, I am going to now explain glycogenesis. I am actually giving overviews because it is a very big topic. So it will be very good for you that just watch this video once and please again read the book just for once. Or if you want to make notes, you can make notes also. But I will be suggesting that making notes, particularly about everything, is very time taking. So the essential parts, I should say, you must first memorize and also do practice by writing, because it is important for exams. Now further on the topic, glycogenesis. Glycogenesis is glycogen synthesis occurring by pathway distinctly different from the reversal of glycogen metabolism. Okay, and the first step is activation of glucose. But over here we can see that I have mentioned another step over here in which glucose is converted into glucose 6-phosphate by action of glucokinase or hexokinase 4 or /hexokinase. Hexokinase is not seen over here because hexokinase is essentially involving glycolysis, not glycolysis, glucose breakdown and glucose metabolism, not glycogen synthesis or glycogenesis. Therefore, I am saying please remember over here, glucokinase is a type of hexokinase, but hexokinase is not seen over here. And the glucose 6-phosphate is produced. Glucose 6-phosphate is acted upon phosphoglucomutase, which converts the glucose 6-phosphate into glucose 1-phosphate. It actually transfers the position of glucose phosphate molecules from 1 to 6 to first position, so producing glucose 1-phosphate, and by the action of UDP, glucose pyrophosphorylase with 1 UTP in addition produces uracil diphosphate glucose or UDP glucose, which is further acted upon by branching enzyme glycogen synthase, and which is responsible for addition of glucose to glycogen molecule. Glycogen actually is the initial part of the glycogen. It is also protein in nature, as I had initially told you that we see limit dextrin from the breakdown of glycogen. The limit dextrin and the glycogen both are different molecules, so please don't mix them, because those are actually one is a part of catabolism, one is a part of anabolism. So in glycogenesis we are seeing catabolism. So glycogen is a part of catabolism, and whereas limit dextrin is a part of anabolism. Okay, okay, and then via the action of glycogen synthase, which add molecules at the alpha 4 linkage, produces linear chains, which is further on branched by the glycogen branching enzyme, which creates alpha 1,6 linkage, and finally creates a glycogen molecule. Glycogen is actually having a core and outer core and an inner core. Actually, I shouldn't be saying that. It could be stated that there is a sphere within a sphere. The inner sphere is glycogen and outer sphere is glucose, made of glucose molecules. Alpha 1,4 linkage, alpha 1,6 linkages can be seen. And there is a block of 6 to 8 glucose molecules. Here, if we count, we can see actually that there are 6 to 8 glucose molecules compound. So it is visible over here. And regulation of glycogen metabolism occurs via it is actually reciprocally regulated. The phosphorylate form of glycogen phosphorylate is active, but glycogen synthase becomes inactive on phosphorylation. The hormonal control by covalent modification and allosteric regulation are interrelated. These hormones act via secondary messengers, cyclic AMP, adenosine monophosphate. Okay, and further on, I will be discussing about the branch formation. We can see over here, which I had initially explained, that UTP glycogen synthase has many cycles which removes UDP, and the glucose molecules are released, and alpha 1,4 link glycogen molecule initially is present. Alpha 1,4 linkages are made, and then branching enzyme transfers 6 to 8 glycogen residues to form a new branch. Alpha 1,6 linkage is seen over. This is alpha 1,6 linkage. Over here it is alpha 1,4, alpha 1,4, alpha 1,4 linkage. Repeat action of glycogen synthase and branching enzyme causes synthesis of glycogen. Okay, so that is for now. I'm actually going to present a new video, or I'm going to further elaborate on other processes related to glycogen synthesis. I will be elaborating on glycogen synthesis and HMP shunt and the related glucose metabolism. The video has been given in a brief for revision purposes. I expect you to please read the books, and please make notes. Notes are really important during the exam times, because books aren't usually helpful. It is better to use notes, self notes, or you may, if you are interested, you can also find notes on internet. And in last, I wish stay motivated, work hard, and all the best for the future. And stay happy and stay safe for the present unpredictable times. Thank you. Bye.